Okay, everybody at home, we are recording, and so go ahead and sign in the chat box. Okay. The other sheet? Okay. Here we go. Okay, that's what I'm trying to get. And I'm trying to make sure that I get people, okay? That. Okay, somebody at home, tell me you can hear me.
Yes.
All right, good, thanks. We don't need this. Okay, so I have some labs that I've graded, right, on paper, and so I'm going to give them back if I see the people. Laura, that's you. Haley, is Haley Bolio here? Melina? No Melina yet. Okay. Jason is not here yet. Kaylee. And Anthony, no, not yet. Okay. All right, well, if you turned a lab in on paper, I've got it. So, except for two of you. Jason. And, oh, wait a minute, there's, no. Wait. Yeah, yours is one that I've still got at the office, okay. Let's see, Lindsay. Okay, for those of you who sent your labs by email, I'll be sending them back by email with little notes on them. It's kind of a pain, but. Okay, I think everybody else is. All right. Okay, a couple of things. I'd like you to remember that I'm expecting to see everybody in the room for the exam on Friday. Now, there's at least one or two people who are not in the state. You need to get with me and remind me that that's an issue. But for those of you who can be here, the exam is here. Unless you have an ODS accommodation, in which case it's over in the library. Second thing, the lab. I'm trying to get all of the dispersion labs graded and handed back to you. I need those as soon as possible if you haven't given them to me. And for today and for the next meeting, if you're not coming today, we're going to be kind of rapidly setting up two labs that will run 2 weeks. And you'll have to pay, somebody will have to come in and water them. But after that, we're going to go over to the science building and analyze the data from the community lab. If you have done it at home and you think you know that you did it right, good on you. You can leave. But if you can't figure out what the heck is going on, today is your day, or whenever you come to lab is your day. We're going to set up these two labs. We're going to walk over to science, sit you down at the computers, and we are going to go through and analyze this. And it will take maybe 5-10 minutes. And once you get the hang of it, it's not that hard. You just have to get the right syntax for the commands in R. Now, this is important because you'll have to actually do one of these labs at home at the end of the semester. And so I want you to sort of have the understanding of how to use this program before I try to make you do that. Okay. So we've already had a number of questions from people about material for exam 2. They've been very good questions. I would say that something that I've repeated to everybody who's come to see me so far is this class on the exams is what you see is what you get. There's nothing tricky about the questions. I keep saying that this stuff is hard enough that I don't have to try to trick you. So it's really important that if you are second guessing yourself, be very sure that your second guess is better than the first guess. Because often in these exams, what hits you first seems like, no, it can't be that simple. Yeah, it's that simple. That's what it is. So don't automatically think that your first guess has to be wrong somehow. Trust your intuition, trust your studying of the exam, and oh yeah, trust the calculations that you do. If you know what you're doing, you should be able to get through the, 4 or 5 calculations in 10 minutes easily. And then the rest of the time you'll have for the 3 responsive questions and the 10 multiple choice. Okay. This is a format that has worked for the last 5 years in a 50-minute exam. So I'm fairly confident that a large majority of you will, in fact, finish. Okay. Now, today we are starting material that will not appear on exam 2. This is the last topic that we will address in population biology. After this, we will move on to talk about species interactions. That is competition, predation, and mutualism. But we've sort of set the groundwork for how we're going to view those interactions. It's particularly important that we went through and got the logistic equation because we'll use that kind of thing in competition and predation models. But today, we're sort of gonna push the envelope and set you up for Dr. Anthony's conservation biology class, because we're going to relax the last set of assumptions about the life table. Okay, remember that when we started this a couple of weeks ago, we said, here are the rates, end of story. And then at some point we started saying, well, okay, we'll mess with these. We'll then allow them to be random variables that have the mean value that we list in the life table, but also have a standard deviation, plus or minus some amount. So a survival rate of 0.5 might be as low as 0 and might be as high as 1. Okay, and that allowed us to model stochastic population growth. Okay, well, the very last assumption that we've got in that life table is that there are only 2 things that can happen. You can either survive or you can die. Well, what if there's a third option? In many real populations, there is more than 2 options. You can do more than just survive to the next time period or die. And so for many real populations, we have a hard time working directly from the life table. We need a somewhat different approach. I am merely going to scratch the surface of this today and show you how this works. We'll do a little bit of calculating just to show you the way that we think of these models. So by way of introduction then, I'll explain the difference between age structure and stage structure in life cycles. You know what, I think I'm going to minimize this. This is taking up too much of the screen. There we go. Okay, then we will talk about an alternate way of writing the life table called the Leslie matrix. Okay, and this is matrix algebra. So it probably strikes you the same way the word calculus does in terms of fear and loathing, but it's actually quite useful, and we are not going to get into the details of matrix algebra. I'm simply going to show you how matrices are used in these more complicated life cycles. Then we'll talk about an alternate formulation called the Lefkovitch matrix, which is the actual thing that conservation biologists and population biologists use to model these complicated populations where there's more than one option of living or dying. And at the end, I'll give you 3 examples of how we have actually used these matrices to understand the dynamics of real populations. So first, let's talk about age structure versus stage structure. All right, well, here's a simple age structured life table. You have 4 ages. You have newborns, that's 0. You have 1-year-olds, and of course, always remember that I say year, this could be days, could be weeks, could be months. It's whatever the organism does. Okay, so we have 4 stages though. Newborns, 1-year-olds, 2-year-olds, and 3-year-olds. Okay, and in the classic age structure for a population, organisms survive from one age to the next. So the rate at which newborns survive to be age 1 is S sub 0. The rate at which 1-year-olds survive to be 2 is S sub 1. And the rate at which 2-year-olds survive to be 3 is S sub 2. Okay, and notice that in this life table, newborns do not reproduce. There is a juvenile stage, so the 1-year-olds don't reproduce, but then 2-year-olds reproduce at the rate of B sub 2, and 3-year-olds reproduce at the rate of B sub 3. So the life table that you derive from this life cycle looks like this at the bottom. And this should look fairly familiar to you by now, since of course you've done 5 life table problems and we went through the life table in lecture. But here are your ages, 0, 1, 2, 3. Here are the numbers of individuals at each age. Here's the survival column and the birth column. Okay, remember, when you reach the age of 3, you reproduce immediately and then you survive or die. So even though the survival rate of 3-year-olds to 4 is 0, they reproduce at the beginning of that interval, right? So that's what the life table looks like for this simple age-structured life cycle. And notice that there's only 2 things that can happen to an individual. That individual could survive at a rate of S, X, or it will die at a rate of MX. And if you don't survive, you die. Well, that's great. That's how we think of us. That's not the way that all organisms work. Here is a more complicated life cycle, and you can't summarize this in a life table very easily. So what's different here? Well, in this life cycle, individuals that are in age class 2 can survive to 3, they can die, or they can stay in this class. Now, how does that make any sense at all? can't be an age class, because the next time step, you'd be a year older, right? So you'd be age 3. So how is it that you can stay in this stage? It's because it's a stage, not an age. So we're talking now not about progressing through life in ages that have a one-way progression from birth to death, but in terms of stages. So instead of talking about 0, 1, 2, and 3-year-olds, we might talk about newborns in stage 0, juveniles in stage 1, and instead of talking about age 2, we might talk about small adults here, and then large adults here. So if we think about it that way, it doesn't matter how old you are. What matters is the stage you are in. So in this life cycle, these small adults could survive to become large adults. They could die, or they could remain small adults. And they would once again have the same birth rate that small adults have. Okay. Over here, the large adults could either die or they can remain large adults. So here we've got the probability of starting in stage 2 and ending in stage 2. And here we've got the probability of starting in stage 3 and ending up in the next time step in stage 3 once again. So now we have some extra outcomes. And the life table doesn't allow us to do this. So we can't use just a standard life table to model this kind of population. And yet these are some of the most interesting and important populations we have. So we must have tools that allow us to model this. So this is kind of the last barrier that we erected when we started working on life tables. First, we said that immigration and emigration didn't matter. And of course, now we're saying they do. Then we said that the values in the life table are constant. And then we said, oh, they can vary. And finally, we're now saying there's more than one way that you can end up in a particular stage. You can live to the next stage, you can die, or you can stay in the same stage. So we have a problem here because we can't use the standard life table. We need a different way of thinking about this. It gets worse because there are a number of species, many of them, in fact, in which there's yet another potential outcome. So in this life cycle, not only can you stay in the same stage, you can actually revert to an earlier stage. Oh dear, there is no way a life table can do that. So what we've got here now is, notice that the 0 and 1 stages are pretty much straightforward, but once you get to stage 2, there are now multiple outcomes. You can survive to become stage 3, which is S2. You can stay in stage 2, which is this probability of 2, 2, beginning in 2 and ending in 2. Or you can revert from stage 2 to stage 1, which is starting in 2 and ending up in 1. Okay, and for purposes of this discussion, I'm talking about making it clear that this is a probability of staying in one stage, and this is the probability of reverting. So that's why I'm using R here. In fact, there's no reason why you need to use different letters. You can just use probabilities and just pay attention to the subscript. There'll always be 2 numbers in the subscript. The first number is where you start. The second number is where you end up. So in this life cycle, you can see that stage 2 has a lot of complexity here. They can survive to be stage 3, they can stay in stage 2, they can die, or they can revert back to stage one. Okay, now we've got some questions. So first I'm going to take Anthea. Okay, so the question is the stage more related to body size or is it more related to behavior? And the answer is it kind of depends on the species. Very frequently, body size is the most important thing that predicts what's going on. Occasionally, it's not. For plants, it's almost always the size. For animals, sometimes it's size, but sometimes it's behavior. Sometimes you have an animal that does not change size very much, but changes behavior. Okay, seeking out a mate versus ignoring the possibility of mating. Okay, second question in the class. Oh yes, I will give you several real life examples, but just for now, let's consider as an example an organism that might actually show both what I call stasis, that is staying in the same stage, and reversion, as well as survival and death. Let's consider a plant that has a cost of reproduction. So that plant grows through seed, seedling, sapling. That's kind of these 1 and 0 and 1 stages because they don't reproduce. And then it enters the reproductive stage. Okay, well, first it's going to be an adult that is not reproducing yet. It's storing up energy to reproduce because there's a cost to reproduction. So then you have an individual that's stored up enough energy to reproduce. So as a result of reproducing, several things could happen. That plant could use so much of its energy that it doesn't have enough to survive, and so it dies. It might have enough energy left to remain in the reproductive state and reproduce again the following year, but not grow bigger. It might have accumulated so much energy that not only can it reproduce now, it can survive to become bigger. And so move into the next stage of, say, a large reproductive individual. Finally, it might have used so much energy that it cannot reproduce the next year, but not so much that it dies. And so it would have to wait a year. It would go back one step to be a non-reproductive adult. So that's typical of organisms that have this kind of structure. There's almost always something like a cost of reproduction. If you have a substantial cost, then you might die as a result of reproducing. You might then use so much energy that you can't reproduce the following year and you go back to a non-reproductive status. You might be able to stay where you are if you've got enough energy, or you might have so much energy that you can get bigger and reproduce more. Okay, and then Dwayne had a question. Oh, that was your question too? Okay. Okay, yeah. At the end, I will show you some 2 animals and 1 plant to illustrate kind of the complexities of these things. And you'll see why I only ask you to recognize things and to do basic calculations and not complicated matrix algebra calculations. You'll see that right away as soon as we start talking about the real life cycles. We're not going to do that. And in fact, in most conservation biology programs, we use computers to just automate this so that we don't have to hurt our brains thinking about how to do the calculations. But then you rely on the program, and you can make terrible, terrible mistakes unless you understand what the program's doing. So just a word of caution there. All right. So here is a really messy population. We can have stasis, that is, you could stay in the same stage. And that I'm using P for that now. But really, just ignore what letter it is. Just look at the subscripts, because that tells you where you start and where you finish. You can also revert to an earlier stage. And you can also grow into the next stage, or you can just die. Okay, so how do we handle something like this? Well, okay. So here's a simple life table written in matrix notation. So this is just your basic plain vanilla life table with all the assumptions intact. And this is exactly the same life table that we saw before. So you have your age column, you have your N column, your survival column, and your birth column. And notice that these are exactly the same as they were in the previous slide. But now if we write this in matrix notation, there's a special way that we handle the variables in a matrix. And so what we define is a vector of ages. So this is where the numbers of individuals are found. They're not actually part of the matrix. The matrix is of the rates. So we've got two rates here. We've got a rate of survival, and we've got a rate of birth. And this is how we write them. So what we have in the top row is the rates that determine how many newborns there will be. In the second row, we have the rates that will determine how many one-year-olds there will be at the next time, how many two-year-olds there will be at the next time, and how many three-year-olds there will be at the next time. And the way that you work with these vectors and matrices is that you take this vector and then you do the transpose thing in Excel. You flip it on its side so that you're multiplying the number at time 0 times this, the number at time 1 times this, the number at times 2 times this, and the number at time 3 times this. And you do that for each row. So to get the number of two-year-olds, you take the number of newborns, multiply it by that, the number of one-year-olds, multiply it by that, number of two-year-olds, multiply it by that, number of three-year-olds, multiply it by that. And that matches the diagram that we've got here. There's only one way to get two-year-olds, and that's for one-year-olds to survive to age 2. So it doesn't matter how many newborns there are, there's no way for them to become two-year-olds. So the rate at which they become two-year-olds is 0. The rate at which one-year-olds become 2 is the number of one-year-olds times that rate. So that's a positive number. There's no way for two-year-olds to stay 2 in this matrix. So it doesn't matter how many there are. It's the number here times 0. And three-year-olds can't go back. So it's the number of three-year-olds times 0. So let's work through it. Do an example here. So n sub 0 times 0 means that there will be 0 newborns in the next time step that come from the current newborns. And that makes sense because look, newborns don't contribute to the next newborn class. One-year-olds also do not contribute. They don't reproduce. So it's n1 times 0. So far you have exactly 0 newborns at the next time step because neither newborns nor one-year-olds reproduce. Now you have two-year-olds and they do reproduce. So they will contribute some newborns to the next time. And it's the rate of the number of two-year-olds times the number of offspring that each one produces. So it's n sub 2 times b sub 2. And then finally, notice that the 3-year-olds also reproduce. Okay, and so let's get rid of the zeros so that we can keep it on one slide. So there are two ways to get newborns. 2-year-olds can reproduce, n2 times b2, and 3-year-olds can reproduce, n3 times b3. You may have newborns in the population, but they don't reproduce. So that's irrelevant. It's however many they are times 0. And that's why we write these values in the matrix. We're being very explicit about the rates. Okay. All right, so how do we get the 1-year-olds? Well, fairly simple. There's only one way. Newborns can survive to be 1-year-old. And they survive at this rate, S sub 0. So the newborns times S sub 0 gives you the number of 1-year-olds. Notice that 1-year-olds cannot stay 1, 2-year-olds can't become 1, and 3-year-olds can't become 1 either. So it's 0, 0, 0. There's only one way to get 1-year-olds, and that's for these guys to survive at this rate. Okay, that works exactly the same way for all the other columns. So to get the number of 2-year-olds, there's only one way to do that. The 1-year-olds have to survive to be 2 at this rate. So newborns times 0, 1-year-olds times S sub 1, 2-year-olds times 0, 3-year-olds times 0. And so that's the value of using this notation. It requires more bookkeeping, but it makes every possible transition explicit. Now, this is called a Leslie matrix, named after the demographer who invented it in the 1940s. A Leslie matrix is simply writing the life table with all the standard assumptions in the form of a matrix and a vector. The vector is always the current population. The matrix is the rates of birth and survival. And you get, by multiplying the vector times the matrix, you get a new vector, which is the number of individuals in each age class at the next time. So the Leslie matrix is simply the life table written this way in order to multiply these things in this way. But as I said, the value is you've made every single possible rate explicit. And notice that some of these rates are 0. Well, in a complex life cycle, those rates will not be 0. There will be a probability that a 2-year-old or a stage 2 individual can become a stage 1 individual again. So you'd have to look in the matrix to find out what value that would be, and it would actually be right here. So currently, there's no way for stage 2 individuals to be stage 1, but if there were, this number here would not be 0, it would be greater than 0. And so you'd multiply the number of newborns times their survival rate. I haven't said anything about a stage 1 individual staying in stage 1, so that would still be 0. But now, if there's a way for these guys to go back, it would be the number of these guys times some non-zero value. And that would be added in to get the new number of individuals in stage 1. So this Leslie matrix is actually kind of a special case of a more general case. In a Leslie matrix, you only have 2 kinds of rates. You have a birth rates and you have survival rates. And notice the survival rates are on a diagonal here, and the birth rates are all on the first row. You see that, you're looking at a Leslie matrix. And it's just a life table. That's all it is. But if we have a more complex life cycle, some of those zeros in the matrix are not going to be zero anymore. They're going to have a positive value. And so here's that first life cycle where there's some probability that stage 2 individuals could stay in stage 2 and some probability that stage 3 individuals could stay in stage 3. Okay, so some of the zeros are now going to be positive values. Well, let's look at how that would affect the matrix. So here's our new matrix of rates. Notice that we have the birth rates still in the first row. We still have this diagonal of survival rates. Yay. But now, look what's not zero anymore. There's now some probability that stage 2 individuals could stay in stage 2. So we multiply this 0 times the number of 0 guys, this times the number of stage 1 individuals, this times the number of stage 2 individuals, and this times the number of stage 3 individuals. So in order to find out how many stage 2 individuals you have in the next time step, you have to figure out what this is, N1 times S1, but you also have to figure out what N2 times this probability is. And notice that the subscript tells you where you're starting and where you're ending. You start in stage 2, you end up in stage 2. So there's now 2 ways to get stage 2 individuals. Stage 1 individuals could survive, N1 times S1, that's right over here. Or stage 2 individuals can just stay in stage 2, and that's N2 times P sub 2, which is right here. And the same thing goes for stage 3. Stage 3 can be gotten by stage 2 individuals surviving or stage 3 individuals staying in the stage rather than dying. So you have stage 1, which can't become stage 3, times 0. Stage 1 times 0, because stage 1 can't become stage 3. You have some individuals of stage 2 surviving to be stage 3. That's this. And you have some individuals of stage 3 not dying and staying in stage 3, which is this, n sub 3 times this probability. If you have non-zero values for these rates that are not on the survival diagonal, then you have what we call a Lefkovitch matrix. It's actually the general case. A Leslie matrix is a special case where there's no possibility of these weird transitions. Okay, in this case, we have 2 non-zero values. We have the non-zero value for 2 staying in stage 2 and for 3 staying in stage 3. What about our other more complicated example? Well, here, I mean, it's just adding in the other rates. So here, stage 2 individuals can go back to stage 1 in addition to the other things that we had. So stage 1 is then given by the new stage 0 times their survival rate. Notice that stage 1 cannot stay in stage 1. So that's 0. So it's this times 0. But now stage 2 can go back. So it's this times the rate of going back. And then stage 3 can't jump over and go back to 1. So that's still 0. So now there are 2 ways to get stage 1. These guys can survive at this rate. These guys can go back at this rate. I have to warn you that I sometimes confuse myself and write the wrong subscripts. Just be alert for that, and always ask the question if you see something that you don't think makes sense. Okay, but now here you can get stage 2 individuals in 3 ways. Stage 1 cannot become stage 2, so that's this times 0. But now stage 1 can survive, so N1 times S1. Stage 2 can stay stage 2, so it's N2 times this. And stage 3 can go back, so it's N3 times this. And here's how you figure out the number of stage 2 individuals. The only thing I'm going to ask you to do with these matrices is to be able to look at a life cycle like this and determine what the matrix should look like. That's one thing. The other thing is that if I give you these numbers in this vector and I give you these rates, I want you to be able to calculate the numbers at the next time step. This is just simple algebra. And we have a question in class. Yes. Right. Okay. So we could write all of these as just P sub something. But since we have already gone to the trouble of talking about the life table in terms of survival columns, and since it actually helps you recognize when you have a Leslie matrix versus a Lefkovich matrix, it's actually kind of useful to retain the S sub 0, S sub 1, S sub 2 notation. Because this is what we used in the life table. We've been using it for 3 weeks now. So if you see anything off this diagonal, then you know it's a Lefkovich matrix. You know there's something complicated about the life cycle. And so I will tend to use S sub 1, S sub 2, S sub 3. But for all the other things, I'll just probably use P sub whatever it is, something, comma, something. Start, comma, finish. All right, let's finish this presentation, which is, complicated enough, by showing you 3 different real-life examples from the conservation literature in which we made use of these matrix formulations. So first, here's sea turtles. These are loggerhead sea turtles, and they live a very long time. And normally when you catch a sea turtle, you have no idea how old it is. So age structure doesn't help us because you can't age them. But what you can do is stage them. So here we have a real study that was published in Ecology, the journal Ecology, that breaks these guys into 7 different stages. First, they're kind of combining eggs and hatchlings together in a single stage. You might wonder why they do that, but I'm not going to go into that. They made the case in setting up their model in the paper. I'm going to just accept that. Okay. Then you have small juveniles. You have large juveniles, so you can tell the difference there. Then you have subadults. So this is a size difference. So you have different size classes. You get the little hatchlings that are maybe as big as a silver dollar. And then you have small juveniles that are maybe this big, medium juveniles, large juveniles, so small juveniles, large juveniles. Subadults would be quite big. They'd be maybe 3 or 4 feet long. So this is a pretty easy thing to do. You can break them into size classes that way. But then you have adults. And you have novice breeders, the ones that have just reached the age of first reproduction, are coming back to the beach for the very first time. Now, the cool thing about that is that if you tag a baby as it leaves the beach, then some years later, it will come back to that same beach. The interesting thing is that when they do come back to the beach, this is their size, this is their approximate age, so somewhere around 18 or 20 years. We can calculate a survivorship for this stage. And here's the fecundity. Here's the number of eggs that they produce in their first year. It's huge. It's 127. on average. Okay, so the interesting thing is when they come back the following year, they only produce about 4 eggs on average. That's because there's a cost to reproduction. This is exactly the kind of situation where a matrix is going to be more useful than a life table. And then after that, if they come back in year 3, 4, 5, we have no way of knowing how many years they're coming back, but they're all big. They live to be almost 60. They have similar survivorship to the other adult stages, and they average about 80 eggs a year. So this is a matrix formulation for the life cycle of sea turtles. Now, what would the life cycle diagram look like? You know, the balls and arrows. Well, I'm going to hand draw you one. And it looks like this. So it doesn't look like the adults that are reproducing go back. They tend to stay in that stage once they reach the mature breeder situation. So there's stasis. So there's a probability that adults will come back as mature breeding adults and breed at this rate, B7. So here's eggs and hatchlings, small juveniles, large juveniles, subadults. Here's your first breeders, and their birth rate is 127. Here's your repeat breeders the following year, their birth rate is only 4. And then here's your adults, and their birth rate is about 80 year after year after year. Okay, so that shows you how those things are related. The matrix formulation is always related to a life cycle that you could look at and draw. So let's look at a second species, and this is polar bears. This is also from ecology. Polar bears have a cost to reproduction, and here is the life cycle that these guys came up with. So here you have stage 1 individuals that are independent from their mother for the first time. They're approximately 2 years old. And that's where we start this. Then we have stage 2 individuals that are 3 years old, stage 3 individuals at 4. They don't really start reproducing until they're about 5. And now here they are. Stage 4 is reproductive individuals, or reproductive potential individuals, because they don't necessarily reproduce. They may not find a mate. They may lose a pregnancy. And so there's some possibility that a non-reproductive adult could wind up staying as a non-reproductive adult. Well, there's stasis. Non-reproductive adults can remain that way, right? They can also breed. And so they can have a cub, right? And if they lose that cub, they could go back to being non-reproductive adults, or they could breed again immediately and have another cub immediately. Or if they don't lose that cub, they could wind up having a year-old hanging out with them. And so once they have a yearling, they can wean that yearling, make it go away, and then they can go back to being non-reproductive for a year and then breed again after an interval of a year. Notice that once you wean a cub, it takes you at least a year to recover from having bred, because there's no way that an adult that has a cub can immediately breed again. There's a cost to reproduction. You have to wait a year before you can breed again. And then here are the cubs, and they go here. Okay, so you can have stasis in 2 places, and you can have reversion in 2 places. If you lose your cub, you can go back to being non-reproductive. If you wean your cub successfully, you definitely go back to being non-reproductive. And Okay, so the question has to do with the current situation with polar bears. Are they breeding less, especially due to global warming? And the answer is yes. Polar bears are basically extinct. They just don't know it yet. I don't see any way of saving polar bears. They're going extinct within your lifetime, as far as I can tell. Okay, the only way to possibly save any of the genetics is for them to interbreed with grizzly bears, which they were derived from grizzly bears about, what, 2 million years ago, something like that. So they're capable of interbreeding. But yeah, these guys are going extinct. Enjoy them while you can. Okay, the last example then is of a plant, and I think you'll see why I don't ask you to do more than some simple calculations and relate a diagram to a matrix. And so here's teasel. This is a really obnoxious plant. Not that there's anything wrong with it, it just has a complicated life cycle. And there are multiple ways in which it can stay in a stage, multiple ways in which it can revert to earlier stages because it has a cost of reproduction. As you can imagine, this took years to figure out. And there just aren't that many species for whom we have these well worked out life cycles and matrix formulations. It's a tremendous amount of work, but let's walk through it very briefly before I let you go. So we start with dormant seeds, and they break dormancy or not, and they can stay dormant. And most of them, as you can see, here's the probability, most of them are going to remain dormant. Some of them can germinate, and they can become small, sort of small plants here. That's the most likely thing. They can become medium plants, or they can actually, if they get a real break, they can become fairly large plants called rosettes. Okay, so this basically is kind of stasis. However, the seeds don't live forever in the soil. If they don't germinate the second year, they're going to die. So these seeds that have stayed dormant for an extra year can only produce small plants. Small plants can stay small. They can grow to be medium-sized plants. They can grow to be big plants. These guys could stay medium. And notice that here's the payoff. Here's flowering. Here's the rate at which these guys can become flowering plants if they're small, the rate at which they can become flowering plants if they're medium, and the rate at which they can become flowering plants if they are large. They didn't put the numbers in, and we don't really care. What's really interesting to me is that once they start flowering, what's the rate of reversion? Notice that flowering plants can't flower in 2 successive years. There's a cost to reproduction. They have to go back. So they can go back to being small plants at this rate, medium plants at that rate, and large plants at that rate. And here's their birth rate. Here's the seeds that each flowering plant produces on average. So this is an example of a really complex life cycle that has multiple complicating factors that there is no way we can use a life table for this. We must make each of these rates explicit. And in order to do that, we use the matrix formulation. So I'm going to give you practice problem for exam 3 to help you sort of visualize how you do the calculations and how you translate a life cycle, not one this complicated, but how you translate a life cycle into a matrix based on the probability that you can go from one stage to another. So you'll have those. I don't expect any of you to do anything with them until after the exam, but you will have them for exam 3. And you'll have a problem on exam 3 that asks you either to write a matrix from a life cycle, or given a matrix and a vector, do some calculations and tell me how many individuals there are in the next time step. Okay. Do we have any other questions, either from home or from the classroom?
Yeah, I had a question.
Okay.
The group that did the lab last week, is the lab report due this Friday?
No, the
Okay.
lab is not due this Friday unless you've actually been able to work it out. If you
No.
have not been able to work it out, we are going to work it out in person today or
Okay.
or Friday or whenever. Okay. All
Okay. Thank you.
right. Anybody else? Okay, off we go. I'll see you in the lab if you're coming today, and I'll see you elsewhere if not. Everybody at home, I'm going to end this. Make sure that you sign in just in case before I end the meeting. 5, 4, 3, 2, 1, done.